Okay, so we've already established this idea in previous lectures about gene families, what a gene family is, and that all of these related copies of genes that we find in genomes are the result of gene duplication. We haven't talked a whole lot about the mechanisms of gene duplication. I'll do a little bit of that in this lecture, but I also want to contrast one other potential source for new genes. Now, by far the most common source of new genes, and even then, maybe you don't classify them as completely new because they're copied versions of each other. But the most common source for expanded genes, having more genes in your genome, is gene duplication. And when genes are originally duplicated, they are most often exact copies of each other. And so you basically just get redundancy of genes when that initial gene duplication event occurs. So in contrast to gene duplication, we also have the potential for what is called de novo evolution of genes, which means genes that come from sequences of DNA that had no transcription, translation. So they were not genes, they were just what sometimes is called junk DNA, or at least non-coding DNA. Now, that's possible, especially in eukaryotes, because we have large chunks of our genome that are non-coding. That doesn't mean they don't have any function at all. They might have structural function, they might have regulatory function, but they are not transcribed and translated into genes. And there are large portions of our genome that don't have much function at all. They're just kind of what you might think of as filler DNA. And so 98.5% of the human genome, for instance, is non-coding DNA. So it is possible that a section of that DNA gains a promoter by a mutation, is then transcribed, and that provides some sort of minimally, it might not be very good at first, but some sort of minimally advantageous change for that organism, and then that becomes a new gene. Now, the odds of that happening are quite low, especially compared to the odds of having something useful or functional when you duplicate a gene. So although de novo evolution was probably very important early on in the evolution of life on Earth, it's much less common now. And by far the vast majority of all genes are resulted from gene duplication event, right? This is this idea of modularism at the genetic level. If we copy what we've already got, and then there's mutations and changes, modifications of that, that's going to be a much more likely mutation to result in something beneficial. So the vast majority of variation that we see between species in their genome and what genes they have comes about via gene duplication. So gene duplication, here are the mechanisms that it can occur. We can have slippage during DNA replication. So enzymes are moving down the DNA chain, making a copy. If they slip and then recopy what they've already copied, we could potentially copy a gene twice. That's probably not super common because genes are very large, and the amount of slippage and then recopying that would have to occur to completely copy an entire gene is not very likely, but it can occur. More commonly, we get these unequal crossing over events that occur during meiosis. And so normally, sister chromosomes line up and then swap parts. If they do that equally, we don't get any duplication. We just get mixing and matching of the allelic variation that is already there. But if they do it unequally, right, if when those chromosomes line up, one part is hanging over the other, and then we get crossing over to occur, we end up with some of the chromosomes that have extra genetic material, like duplicated genes, and then some of the chromosomes that are completely missing that genetic material. The chromosomes without the genetic material are probably going to be a very negative mutation and be removed by natural selection, but the ones with extra genes, extra genetic material, could very well and often are beneficial. So all of these things can occur via unequal crossing over events. We could get a chromosome missing a gene. If non-homologous chromosomes lined up and then swapped parts, we could end up with a gene from a very different part of the genome being moved in, and those are somewhat common events. All of these are mutations. If they're negative, they'll be removed by natural selection, and we would never see them. But enough of them are positive, and genome recombination and genome rearrangement is frequent enough that we can have even very closely related species that have very different arrangements of their genomes. Okay, so these are not uncommon events, but the vast majority of the time they're going to be a negative mutation removed by natural selection. We could have inversion where we have the order of the genes that's changed, and again, via unequal crossing over, most commonly we could end up with many copies of the same gene, usually 2 to start with, but then if it happened again, we could end up with 4, and we can get large regions with many different genes that are all duplicated. And all of these have played a role in the evolution of the Hox genes and other major toolkit genes. So all of this genetic material can result in extra genes in some organisms, and if that's advantageous, that will eventually spread, and soon the entire species will be carrying those extra duplicated genes. This is very, very common, and I could have picked from thousands of examples. This is just one I found online that was nice and easy. Hemoglobin, which is an oxygen-carrying molecule, is present in many animals. In some arthropods, they have also copies. And Daphnia, which is a freshwater crustacean, very common in ponds, you can find them here quite easily, has 8 different copies, all right next to each other on the genome. So fairly frequent duplication. In fact, Daphnia's has many, many extra copies of many of the genes. So in fact, one of the other ones, in fact, let's add a third one here, which is maybe kind of obvious, but bears mentioning. In addition to unequal crossing over, which can copy one gene or maybe a group of genes that are all kind of close to each other on a chromosome, a third very important one, which again kind of goes without saying, but is genome duplication. So of course, if you have a mistake where you copy your entire genome, you're going to end up with extra copies of every single gene in your genome. So genome duplication, or even we could maybe even put chromosomal duplication, right? Many chromosomal duplication events are fatal and of course would be removed by natural selection, but genome, let's just put genome/chromosomal duplication events are also of course going to result in extra genes in the genome. Okay, and many of these are not kept, but sometimes they are. And so with this example of Daphnia, the most reasonable explanation for this is that we had a number of chromosomal duplication events that led to this. Now, if this was the only gene that we saw duplicated and none of the other genes were, then that might say, oh, well, let's look at unequal crossing over events. That might explain why this gene has so many copies and other genes don't. Okay, but it happens by very many mechanisms. It's a very common way, in fact, by far the most common way to get extra genes in the genome. Now, once this occurs and we have a number of genes, maybe with slight variations as new mutations occur that are right next to each other, there's a little bit of what we might think of as a snowball effect or domino effect, where after one gene duplication event, it's more easy to have multiple gene duplication events, maybe even within a gene as they've marked here. But those chromosomes, when they line up during meiosis, they match up based on similarity. And so if you have areas of the genome that are very similar, even identical to each other, and right next to each other, then that causes more misalignment. So unequal crossing over kind of has a little bit of a snowball effect where you're going to have more likely, it's still a very low number, but more likely than it was before to have more unequal crossing over events. And so often we will see within a group, once a gene duplication event occurs, that there's more and more of a tendency for those to get even more copies of that gene and end up with a large superfamily of genes. And of course, the Hox genes are one of those gene families that has gone through these gene duplication events. So I've got the ancestor of the canonical Hox genes here, Hox genes from different insects with them kind of highlighted. This was a data set I put together several years ago. And notice that some of them we can see are fairly closely related to one another. Others, maybe it's a little trickier to figure out. There's still a lot of work that needs to be done. But all of these Hox genes have resulted from gene duplication events, probably most commonly through unequal crossing over. And so we end up with this big gene family of critical control genes that could change the way an organism develops and physically change quite a bit about the way that they look, the way that they develop. So it's a really cool group to look at and to study if you want to understand the evolution of animal form. And so, of course, we could classify all of these as they're all homologous genes because they all have a common gene ancestor, but we could classify them as paralogs, meaning two genes that go back to a gene duplication event to find their common ancestor, or orthologs, which are two genes that we trace them back to a single species that had one copy of that gene. And so our nomenclature, the way that we name genes, matches this, where we would call all the genes that are all paralogous, we would call them a gene family. But orthologous genes all get the same gene name, or at least get recognized as orthologs if we accidentally give them different names to begin with. like the eyeless PAX6 example that we've talked about earlier. So each of these colored circles represents orthologous copies of the same gene in different species. And then those all grouped together in the common gene ancestors, these would be gene duplication events, the ones that are outside of these highlighted orthologous groups. Now, hopefully you understood most of that, but the key is if we now have many genes in the same gene family that are all similar enough, we can align them and do an analysis and do a gene phylogeny that represents both speciation events and ones that represent duplication events. So here's an interesting one in engrailed. Now in arthropods, engrailed is a segmentation gene. It's also an important developmental gene in the vertebrates, but not in the same role. It doesn't play that early segmentation role. But anyway, there are multiple copies and they have gained differential function in different species. And so when we put all the genes together and align them and then look at the species phylogeny, sometimes what we will see is the species history represented multiple times in our gene phylogeny. So humans closely related to mouse, closely related to chickens. Engrailed 2 shows that same pattern as engrailed 1, and then their closest relatives, or the outgroup for the vertebrate engrailed genes, are zebrafish genes. And notice this one was called 3, really should be probably called 2b. But anyway, the asterisks here represent duplication events. And this is an ancient, probably we should put 2 asterisks here because there are 2 duplication events leading to the 3 copies of the engrailed gene that we have in vertebrates. And in fish, I'm sorry, it's a single one. We actually should probably, oh no, it's still 2 copies, so they did it right. An asterisk here, which represents the original gene duplication event for 2 copies, and then 2 separate duplication events leading to 4 copies in the fish. Okay, and so this is just simply a more complicated, and I don't care that you're able to do this, but recognize that we can make phylogenies of gene families and then use the species relationships and the gene relationships to map gene duplication events onto the history of organisms. And so in a more simplified version where we're only showing relationships of the species without all of the genes, we can map it onto. This is where we should put 2 gene duplication events to lead to 4. Or I guess I'd take it back. It could actually be just 1 gene duplication event if we get a double here and then a duplication of that entire region, right? And so this maybe we should put a question mark. In fact, let's do that. I'm going to insert just a text box here. We'll just do a little question mark. So realize the reason I'm doing this is to get to 4 copies of this gene. We'll just put it right there. How's that? To get to 4 copies of this gene, if we duplicated the gene originally and then duplicated both of those copies in 1 mutational event, we would end up with 4. Now, alternatively, we could duplicate the gene to 2 and then have a duplication of 1 of those copies to 3 total, and then a duplication of the other one later on. So we could have separate gene duplication events here to get to 4, or we could have just 1 big duplication of the already duplicated gene to get to 4. So that might need to, and we might need to do a phylogeny of the genes to figure out. Now, in fact, if we look back, the way they have it mapped here, they have it mapped as 2 separate gene duplication events. So really, we should put 2 bars here representing 2 duplication events at different times in the zebrafish. And that's based on the analysis of the gene relationships. So the end result is an ancestral, only 1 single copy of this engrailed gene, but now all of the higher vertebrates, the amniotes, have 2 copies. And the zebrafish, right, the fish, and we would maybe need to do other fish to see if it was just in the zebrafish lineage or more anciently in the ancestor of all fish, but have 4 copies. We would assume it's a fish trait until we did more sampling to see if it was something more recent and specific to just the zebrafish. All right, so you can see how we get genetic diversity via these mechanisms. And so the end result is we often have these gene families that are still very, very close to one another. And in the Hox genes, they have been maintained close to one another by natural selection. And so we have a canonical group of Hox genes that are maintained there by selection, but other Hox genes, either more ancient or that have been rearranged and shuffled to different parts of the genomes, that also have features of these Hox genes, and so they're related to them, but not classified as part of the Hox complex. Okay, and so these are different chromosomes in the human, and humans not only have this duplication occurring within the Hox complex itself, but then the entire complex was duplicated. And we'll talk about that here in our very next lecture when we look at some really critical, important events that have occurred here. So gene duplication increases diversity, increases the number of genes that you have, and basically can expand all of the genes, but it's especially important for EvoDevo looking at the expansion of the toolkit and then what that allows you to do developmentally. Okay. Now, in a general sense, we're going to classify 4 possible outcomes of a gene once it's been duplicated. Probably the most common outcome for a duplicated gene is that we lose one copy of the gene. Now, when I say lose, it doesn't mean it's completely deleted, that we wouldn't see it in the genome anymore, although that's possible. But more frequently, what occurs is it's just no longer a functional gene. It's no longer transcribed. And so when that occurs, when the DNA sequence is still there, but it's not a gene, and so we lose it as a gene, we would call it a pseudogene. A pseudogene is a duplicated gene that is no longer transcribed and translated, so it's just become part of the non-coding DNA of that species. And in the globin family in humans, we actually have 2 pseudogenes that are found within the 5 functional copies of the hemoglobin in humans. Okay, so we can identify pseudogenes based on their sequence similarity, but if we recognize or find out that they're no longer being used, they're not transcribed and translated, then they are classified as pseudogenes. And this is probably the most common outcome, because when we get a duplicated gene, it's redundancy. And so mutations to one of those genes are not going to be very negative because you still have a functional copy. And so the reason pseudogene conversion to a pseudogene is so common is because of that redundancy of function that is generated by the initial gene duplication. Now, along those same lines, if one of the genes mutated and became different, representing these black lines, but still controlled the same way, if that happened, we would get new function to that gene. Okay, now there's a term for this, and I'll put it in here. I like this term. It's big and complex, but if you know a little bit of Latin or even can recognize related words in English, it's called neofunctionalization. So when we get a copied gene that diverges in function, there we go, that's the way I want to do it. Let's group these together. When I get a copied gene that diverges in function, that is called neofunctionalization. And so same thing applies here, right? We have duplicated genes, we have redundancy initially. So when this one mutates and starts to do something new, if it's beneficial and not just, making it so it's no longer functional, if it's beneficial, it can be kept very, very easily because we don't lose the original function. We have a conserved version of the original gene that can still do whatever it was, and if that's important, right? And now we have a second function. So not only do we get redundancy when genes duplicated, but we get the possibility for new function originating without losing the old function. So it's a very flexible and useful mechanism for new function at the genetic level. Okay? And so there are some evolutionary things that I just want to touch on briefly. This is an evolution class, but there are many evolution concepts we don't delve into, but this is important. And you look at it and you say, I don't maybe remember exactly. Hopefully you've already taken an evolution course. I think most of you have. But let's define stabilizing selection in case you've forgotten what it is or you haven't had that evolution class. And then we'll look at this concept. Stabilizing selection is natural selection that maintains the function of a gene. So if a gene is very, very critical to survival, we are going to see stabilizing selection. This is sometimes also referred to as purifying selection. And in fact, I think more commonly we should probably, let's change this and just refer to it as purifying selection. So purifying selection means that changes that change a gene sequence and lead to new protein sequence are not going to be kept very often because they'll change the function of that very important gene. But changes that are neutral, right, that are synonymous, can be kept because they don't change the function of the gene. And so we can actually measure this, and there are methodologies and comparisons and analyses that can be done to measure whether it's there or not. But I just want you to understand the concept. And so this purifying selection and neutral theory are really connected to one another, where neutral mutations are allowed to stay, but changes that change the protein sequence are not allowed to stay. And so they're selected out by natural selection. Okay, so those two things are connected, and we can see that in action here where this gene would be under purifying selection, but because we have released it from so much constraint, because we have redundant copy, this one can evolve and change and kind of explore possible, random mutations followed by natural selection, but explore possible advantages that wouldn't be possible if we only had one copy. So duplication releases some of the constraint, the purifying selection, on really important genes, or at least on one copy of the now duplicated really important gene. Now, in addition, this one's very, very similar. We could have maintenance of the function of the gene, but new regulation. So one of them is turned on in new tissues, but the same basic function of the gene, because we still have both of them under purifying selection, with the just being turned on now in different tissues. And this might lead to pleiotropy of this gene, where we now have one gene with, I'm sorry, I take that back. This is not pleiotropy because we have a duplicated version of the gene. But we could have divergence of regulation with maintenance of the same basic function. So this, maybe a good example of this would be the PAX6 and ILS gene, where basically they have maintained the same exact function, but they're regulated differently. And so if I took a mouse gene with all of its regulatory things and put it into a fruit fly, it would totally freak out and wouldn't be useful at all. But if I took only the mouse gene and then put it on here where it has all of the fruit fly regulation, then it's, because of its conserved function, it could still do what it needed to do. Okay, so that's just an example, but if that occurred in the same species, we'd have duplicated genes with that tendency. And then finally, this occurs also where we could have long-term stabilizing / purifying selection on both copies. And this occurs when there's an advantage to having two copies. And this can occur if we've got some sort of dosage advantage, where if you can make extra amounts of this exact same gene, it's better than it was before, but we still need it to a very conserved function. So long-term conservation of function of both copies is the fourth possibility that it can occur when we have a duplicated gene. Now, this one's not super really important for evo-devo. We're really more interested in the ones here that are listed as 2 and 3, right? When a gene gains brand new function, either through changes to the gene itself, or it gains new regulation so it can do something different, that one of those copies can do something different than the ancestor could do. And when that occurs, we have the potential for developmental novelty, maybe even dramatic changes to the organism that might result in an organism or a group of organisms that can do something completely new that its ancestor could never do